We are doing a massive deep dive into Chapter 42, Regulation of Respiration. And let me tell you, having read through this source material, this isn't just about lungs inflating and deflating like a pair of bellows. This is about a highly sophisticated, a neurogenic system located deep in the brainstem.
That's right. It is a control system that adjusts ventilation, how much air you move, to match the body's demands with incredible precision. And when we say precision, we aren't using that word lightly.
Oh.
The system is designed so that your blood oxygen and your carbon dioxide levels hardly change at all, even when you're transitioning from sitting on the couch to doing heavy exercise.
That was the first thing that really blew my mind in the reading. You'd think, logic would dictate that if you start running, your oxygen levels would tank and your CO2 would spike, and then the brain would say, oh no, emergency, and you'd start breathing harder to catch up. But that's not what happens, is it?
No, it's not. Not at all. If that were the case, you'd be passing out every time you ran for a bus. But the system is predictive. It's way, way smarter than a simple reaction loop.
So our mission today is to explain every line of these control systems. We are going to go long today because we really need to unpack this. We're going to look at the hardware, the neurons in the brainstem.
The control room.
Exactly. Then we'll look at the software, the chemical sensors that are actually driving the whole thing. We'll look at the stress tests like high altitude and exercise, and we will definitely talk about what happens when the system glitches, like in sleep apnea or that terrifying thing called Cheyne-Stokes breathing.
And we're going to explain the chemistry in a way that actually makes sense, specifically regarding why carbon dioxide is actually the boss, not oxygen.
Which is a plot twist I did not see coming. So buckle up. We are going to the control room. Let's start with the geography. When we talk about the respiratory center, where exactly are we looking in the brain?
We are looking at the brainstem, specifically two areas called the medulla oblongata and the pons. Now, for those who aren't neuroanatomists, the brainstem is, the stalk at the base of your brain that connects to the spinal cord. It controls all the primal stuff: heartbeat, consciousness, breathing. And the respiratory center isn't just one spot. It's composed of several groups of neurons located bilaterally, meaning on both sides of the brainstem.
The source material breaks this down into three major collections of neurons. I want to walk through these one by one because they each have a distinct personality, a distinct job. The first one mentioned, and arguably the most critical, is the dorsal respiratory group.
Correct. The dorsal respiratory group is located in the dorsal, or the back part of the medulla. This group is the rhythm maker. Its primary function is inspiration. It tells you to breathe in.
Okay, rhythm maker. That implies it's setting the tempo for the whole show. Now, looking at the anatomy here, it says most of these neurons are located within something called the NTS, the nucleus of the tractus solitarius.
Right.
Now, that sounds like a location in a fantasy novel, but why is that specific location so significant for breathing?
Think of the NTS as a grand central station for sensory information. It is the sensory termination point for two of the most important nerves in the body, the vagus nerve and the glossopharyngeal nerve.
The big data cables.
That's a perfect analogy, exactly. These nerves are the information highways, bringing signals from outside the brain. They bring data from peripheral chemoreceptors, which we'll talk about, from baroreceptors, which measure pressure, and from receptors in the lungs themselves. So the dorsal respiratory group is sitting right where all the sensory data comes in. It's not in an ivory tower. It's on the trading floor. It's getting live data.
So it's the hub. It's getting all the reports from the field. Oxygen is low. Lungs are stretched. Blood pressure is high. But here is the mystery mentioned in the text that I want you to clarify, because this is wild. The text says, even if you cut all those nerves, if you cut the vagus, cut the glossopharyngeal, essentially blinding the brainstem to the outside world, what happens?
It keeps firing. The dorsal respiratory group will still emit repetitive bursts of inspiratory action potentials.
That is just, it's hard to wrap my head around that. So the rhythm is built in. It's not just reacting to the world, it has an internal beat.
Exactly. It has an intrinsic rhythm. We don't know the exact mechanism of those repetitive discharges in humans. It's still a bit of a black box, but it's a fundamental network. In primitive animals, they've found these simple neural networks where one set of neurons excites a second set, which then, after a delay, inhibits the first set. And that creates a continuous on-off loop.
Like a biological oscillator.
Precisely. It's likely something similar in humans. It's a biological clock that ticks every few seconds for your entire life without you ever winding it.
Okay, so the dorsal group says breathe in. But this isn't a digital signal, is it? It doesn't just send a lightning bolt to the diaphragm. The text describes something called a ramp signal.
Yes, and this is a beautiful piece of biological engineering. Imagine if the nerve signal were a sudden burst like flipping a light switch on. Your diaphragm is a big muscle. If it contracted instantly and violently, you'd have an inspiratory gasp.
Like a hiccup or a startle reflex.
Or worse, it would be jerky, uncomfortable, and actually inefficient for filling the lungs with air. You want smooth, laminar airflow. So instead of a burst, the signal begins weakly and increases steadily, like a ramp, for about 2 seconds.
So it's like turning up a dimmer switch on a light rather than flipping the toggle.
Precisely. That is a perfect analogy. This steady increase causes the diaphragm to contract progressively, creating a smooth vacuum in the chest. The lungs fill smoothly and steadily. Then, after about 2 seconds, the signal abruptly ceases.
Just cuts off.
Just stops. For about 3 seconds.
And that cessation, that's the exhale.
Yes. That turns off the excitation of the diaphragm. Now, this is important. Quiet expiration is passive. You stop pulling, and the rubber band snaps back. The natural elastic recoil of the lungs and the chest wall is what pushes the air out.
So breathing out, at least when we're just sitting here resting, is just the system turning off. It's not an active push.
Direct. Quiet expiration is purely letting go. You're not actively doing anything.
Now, the text mentions that we can tweak this ramp signal in 2 ways to change how we breathe. This seems like the control knobs for the entire system.
Right. Think of it as a synthesizer. First, you can control the rate of increase of the ramp. If the ramp goes up steeper and faster, the lungs fill more rapidly. You're taking a fast, hard breath.
Like when you're startled.
Exactly. But second, and this is the usual method for controlling the rate of breathing, you can control the limiting point.
The limiting point meaning when you decide to cut the signal off.
Exactly. If you cut the ramp off early, say after 1 second instead of 2, the breath is shorter and more shallow. If the breath is shorter, you can fit more of them in a minute. Your respiratory rate goes up.
This brings us to the second major group of neurons, the pneumotaxic center. I love that name. It sounds like a piece of industrial machinery from the 19th century, the pneumotaxic center.
It does sound mechanical, doesn't it? It's located dorsally in the superior pons, so it's slightly higher up the brainstem than the medulla. Its primary function is to be the switch-off mechanism.
The timer.
The timer,
Yeah.
exactly. It transmits signals down to the inspiratory area to cut that ramp short.
So if the dorsal group is the gas pedal, the pneumotaxic center is the brake.
In a sense, yes, or more accurately, it's the governor on the engine. It limits the duration of inspiration. Let's look at the extremes. If the pneumotaxic signal is very strong, it might cut the ramp off after only half a second.
So a tiny little puff of air.
A tiny puff. You only get a little air in, but because the cycle is so much shorter, your breathing rate can increase to maybe 30 or 40 breaths per minute.
Short, shallow panting, like a dog on a hot day.
Exactly. Conversely, if that pneumotaxic signal is weak or absent, the ramp continues for 5 seconds or more. You take a huge, deep sighing breath, filling the lungs completely. But because it takes so long, your rate might drop to maybe 3 to 5 breaths per minute.
So the key takeaway here is the pneumotaxic center controls the rate of breathing by limiting the depth. You can't breathe fast and deep at the same time, at least not efficiently, because the very mechanism for increasing the speed is cutting the depth short.
That's a great way to put it. There's a trade-off.
Okay, we've got the rhythm maker, the dorsal group, and we've got the switch, the pneumotaxic center. There's one more group in this control room, the ventral respiratory group.
Right. The ventral group is located anterior and lateral to the dorsal group. The interesting thing about this group, the main thing to remember, is that during normal, quiet breathing, it is totally inactive.
It's sleeping on the job.
It's on standby. During quiet breathing, the dorsal group and the pneumotaxic center handle everything. But when the respiratory drive becomes greater than normal, think heavy exercise, running upstairs signals spill over from the dorsal group into the ventral group. It's like an overflow valve.
So this is the overdrive system, the afterburner.
Exactly. The ventral group contributes to both inspiration and expiration. But crucially, it provides powerful expiratory signals to the abdominal muscles. Remember we said expiration is normally passive.
Right, the rubber band just snapping back.
Well, when you're running a marathon, you don't have time for passive recoil. You need to force the air out to make room for new air immediately. The ventral group activates the abs, they contract, and you squeeze the air out quickly and forcefully.
So that feeling when you're exercising and you can feel your stomach muscles engaging with every exhale.
Yeah.
That's the ventral group waking up and taking charge.
That is the ventral respiratory group coming online. It turns expiration from a passive act into an active, powerful one.
Okay, so to summarize the hardware before we move on, dorsal group is for the basic in and out rhythm. Pneumotaxic center controls how long you breathe in, which in turn sets the speed. And the ventral group is the backup generator that kicks in when you need to breathe hard and fast, specifically recruiting the abs for a forceful exhale.
That is the perfect summary of the central neural layout.
All right, before we get to the chemistry, which is where the real magic happens, I think, there is a safety valve mentioned in the text called the Herring-Breuer reflex. This involves receptors in the lungs themselves, not in the brain.
Right. This is a feedback loop coming from the periphery. In the muscular walls, the bronchi, and the bronchioles, there are stretch receptors. When the lungs inflate, these receptors get stretched,
Makes sense.
and they send signals up the vagus nerve to the dorsal respiratory group in the brainstem.
And what do they say when they call home?
They say stop. When the lungs become overstretched, these receptors activate a feedback response that switches off the inspiratory ramp. It's very similar to what the pneumotaxic center does, but the signal is coming from the lungs themselves, not from higher up in the brain.
So is this regulating our breathing right now, as I'm sitting here talking to you?
Probably not, no. In adult humans, this reflex has a pretty high threshold. It usually doesn't kick in until the tidal volume, that's the amount of air you move in a single breath, is greater than 1.5 liters.
And a normal breath is what, about half a liter?
About that, yes. So we're talking about 3 times a normal breath.
So I really have to be gasping for air, taking a massive deep breath for this to trigger.
Yes. It's a protective mechanism to prevent excess inflation and lung damage. It's like a circuit breaker in your house. It's not there to dim the lights. It's there to stop the house from burning down if there's a huge power surge. It's a safety feature, not a primary controller of normal breathing. Okay, that's a great distinction. Let's move to section 3, the chemistry lab. This is where the source material gets into the why. What is this whole elaborate system actually trying to control? The ultimate goal is simple: to maintain proper concentrations of 3 things in the tissues: oxygen, which we call O2, carbon dioxide, CO2, and hydrogen ions, H plus L PEPA. But, and this is the big aha moment for most people, they do not all control the brain equally. Not even close.
This is the part that always surprises me. We think of breathing as getting oxygen. We equate breath with oxygen. I'm out of breath means I need more oxygen. But the text says oxygen has almost no direct effect on the respiratory center in the brain.
That is absolutely correct. The respiratory center in the brainstem is not directly sensitive to oxygen. You could have dangerously low oxygen in the fluid surrounding those neurons, and the respiratory center itself wouldn't necessarily ramp up based on that alone. The direct drivers, the things that really push the buttons, are CO2 and hydrogen ions.
Wait, play that back. The thing we need most to survive oxygen isn't the thing running the show. That seems like a design flaw.
It seems like one at first glance, but it's actually brilliant evolutionary engineering. Under normal conditions, our oxygen levels are usually sufficient because of how hemoglobin in our red blood cells works. It's a fantastic buffer. But our waste product CO2 is constantly being produced by every cell in our body, and CO2 is dangerous if it builds up because it changes the pH of our body.
It makes us acidic.
It makes us acidic, and enzymes stop working when the pH is wrong. So the body prioritizes getting rid of the trash over constantly checking the fuel gauge, because the fuel tank is usually pretty full.
Okay, let's talk about that mechanism. There's a specific chemosensitive area mentioned.
Yes, this is a very specific patch of neurons located just a fraction of a millimeter beneath the ventral surface of the medulla. These neurons are highly sensitive to the chemical composition of the fluid around them. Specifically, they are excited by hydrogen ions.
Hydrogen ion, that's essentially acidity, right? More H plus means more acidic.
Exactly. Hydrogen ion concentration means high acidity. When these sensor neurons sense that acid, they get excited and they scream at the respiratory center, "Breathe harder, get this acid out of here."
But here's the puzzle introduced in the text, and I need you to walk me through this slowly because it's a bit of a mind bender. Hydrogen ions do not easily cross the blood-brain barrier.
Correct.
That barrier is like the bouncer for the brain's nightclub. It protects the brain from charged particles. So if I have acid in my blood, the brain shouldn't know about it. The bouncer should stop it at the door.
That is the blood-brain barrier puzzle. You are absolutely right. Hydrogen ions, H plus A, are charged. They bounce off the barrier. They can't get in. But carbon dioxide, CO2, is a gas. It's not charged. It's neutral. It passes through that barrier as if it doesn't even exist. It flows right through.
So CO2 acts as a Trojan horse.
In a way, yes. Let's trace the path. You have high CO2 in your blood from, say, holding your breath. That CO2 crosses the blood-brain barrier into the cerebrospinal fluid and the brain tissue itself.
Okay, it's inside now.
Once it is inside the brain fluid, it reacts with the water that's everywhere in our bodies.
Basic chemistry time. CO2 plus water, H2O equals.
Carbonic acid, H2CO3. And that acid is unstable, so it immediately dissociates. It breaks apart into hydrogen ions, H plus Ar, and bicarbonate ions.
So the CO2 sneaks in and then transforms into the hydrogen ions that the sensor can actually feel.
Precisely. The brain doesn't sense the CO2 in the blood directly. It senses the hydrogen ions that the CO2 creates inside the brain. So paradoxically, CO2 is the potent driver of breathing, but only because it transforms into hydrogen ions once it has crossed the barrier.
That is such a cool workaround. It explains why changes in blood CO2 have such a massive, immediate, acute effect on your breathing. If your CO2 goes up, your brain becomes acidic locally, and you start panting to blow off that CO2.
Right.
But the text mentions an acclimatization or fade effect. If I have high CO2 for a long time, the effect wears off. Why? Does the brain just get tired of screaming and give up?
No, the body adapts. This is crucial for people with chronic lung diseases like COPD who live with high CO2 levels. The stimulation by CO2 is very strong for the first few hours. But over 1 to 2 days, that powerful effect declines to about 1/5 of the initial effect.
So what changes? How does the body do that?
The kidneys get involved. The kidneys are the master regulators of the body's long-term pH balance. When they see chronic acidity in the blood, they start to increase the level of bicarbonate, HCO3, in the blood. Bicarbonate is a base. It acts like a sponge for acid. It binds with hydrogen ions to neutralize them.
It's like the body's own natural Tums or baking soda.
Exactly. This extra bicarbonate slowly diffuses through the blood-brain barrier into the brain fluid, and it binds up those excess hydrogen ions that are sitting around the chemosensitive area.
So the kidneys send in a cleanup crew to neutralize the acid in the brain.
Exactly. So even though the CO2 level is still high, the hydrogen ion concentration around the sensor drops back toward normal. The sensor stops screaming so loudly. That's why a change in blood CO2 is a potent acute controller, but a weak chronic controller. The system adapts.
That is fascinating. It explains why someone with emphysema might have sky-high CO2 levels but isn't hyperventilating constantly. Their brain has buffered it out.
Correct. But this creates a new problem, a very serious one. If the brain is now ignoring the CO2, what is left to drive their breathing?
Well, if the brain doesn't care about oxygen directly, we must have a sensor somewhere that does. Because if I hold my breath, eventually I need oxygen. Something has to care about oxygen.
We do. And that brings us to section 4. The oxygen sensor, or more formally, the peripheral chemoreceptors. Since the brainstem doesn't sense oxygen directly, the body puts sensors outside the brain, in the bloodstream.
Okay, where are they?
They are in 2 very strategic locations. The carotid bodies, which are located in the neck at the bifurcations, the split of the common carotid arteries that feed the brain.
Prime real estate. They're tasting the blood right before it goes to the CEO.
The best real estate. And the aortic bodies, which are located along the arch of the aorta, right as freshly oxygenated blood leaves the heart. And these little bodies are special. The text notes their blood flow is extreme.
Yeah, it says 20 times their own weight per minute. That's a huge amount of flow.
It's massive. And what that means is, look, most tissues extract a lot of oxygen from the blood. The blood leaving the tissue is venous. It's depleted. But because the flow is so fast in these chemoreceptor bodies, they don't have time to extract much oxygen. They're constantly being bathed in fresh arterial blood.
So they aren't sampling used blood. They are tasting the vintage straight from the cask.
Exactly. They are sensing the true arterial oxygen level.
And what triggers them? What makes them fire?
They are exquisitely sensitive to low oxygen. But, and this is so important, they are a rescue system. They are an emergency system. Under normal conditions, where your arterial oxygen pressure, your PaO2, is above 60 to 70 millimeters of mercury, oxygen plays almost no role in regulating your breathing.
Wait, really? So right now, sitting here, my oxygen levels are fine, probably around 95 or 100 millimeter each G. So these sensors are basically quiet.
They're sending a low baseline signal, but they aren't driving the bus. Your CO2 level is driving the bus. But if your oxygen falls below 60 millimeter Hg, which is quite low, that's called the ambulance territory for most people, the impulse rate from these sensors shoots up rapidly. It's an exponential curve. The lower the oxygen, the faster they fire.
The source material goes into a deep dive on the mechanism inside these receptors. It talks about the glomus cells. This is in figure 42 to 5 and 6. I want to walk through this cellular Rube Goldberg machine because it's amazing that we know this level of detail.
It is absolutely fascinating. These chemoreceptors contain these special glandular-like cells called glomus cells. And here is the sequence of events when oxygen gets low.
Okay, lay it out.
Step 1. Low oxygen in the blood reaches the glomus cell. This causes very specific oxygen-sensitive potassium channels in the cell membrane to close.
Okay, potassium channels close. Usually potassium leaks out of cells to keep the inside negative, right?
Right, that's what maintains the resting potential. So if you close the door that lets the positive potassium ions out, what happens?
The positive charge builds up inside the cell.
Exactly. Step 2. The cell depolarizes. The voltage across the membrane changes. It becomes less negative.
This electrical change, this depolarization, opens up a different set of channels. Voltage-gated calcium channels, calcium ions, Cal plus I, rush into the cell.
Calcium influx. In biology, calcium rushing into a cell almost always means action.
It does. It's the universal trigger. Step 4. The increased calcium inside the cell causes it to release a neurotransmitter. The text suggests it could be ATP or dopamine or acetylcholine. We're still working out the details. This neurotransmitter then diffuses across a tiny gap and stimulates the nerve endings connected to the glomus cell.
And that nerve sends the signal up to the brainstem.
It sends an alarm signal up the glossopharyngeal or vagus nerve to the brainstem saying, hey, we are running out of air down here. Do something.
And this signal can be powerful. It can really drive breathing hard if oxygen drops low enough. It can even overtake the central CO2 response.
It can. This interaction is shown beautifully in figure 42-8. It shows that these things, CO2, pH, and oxygen, don't work in isolation. They're a team.
What does the graph show?
The graph shows that if your oxygen is very low, the system becomes much more sensitive to changes in CO2. It's like the body is saying, okay, oxygen is already scarce, so we really can't afford to have any extra CO2 in here. We need to breathe it out immediately. The two signals potentiate each other.
This brings us perfectly to a real-world scenario where these systems come into direct conflict. Mountain climbing. Section 5 covers chronic low oxygen.
This is a classic physiological dilemma, a real challenge for the body. Imagine a climber ascending a mountain slowly over several days. The air is thin, oxygen is low.
Okay, so the peripheral sensors, the carotid bodies, they start screaming, low oxygen, breathe deeper, breathe faster.
Right, and the climber does, their ventilation increases, but, and here's the conflict, when you breathe deeper and faster, what happens to your CO2?
You blow it off, your CO2 level drops.
And we just learned that low CO2 tells the central chemosensitive area in the brain stop breathing. It causes what we call respiratory alkalosis.
So the oxygen sensor is flooring the gas pedal, but the central CO2 sensor is slamming on the brakes.
Exactly. The brainstem detects the low CO2, the alkalosis, and it inhibits the respiratory drive. This braking mechanism is powerful. It usually limits the increase in ventilation to maybe 70% more than normal, even though the body desperately needs much more oxygen than that.
So you're stuck in this physiological tug of war. But climbers do get used to it. They can summit Everest. How do they break the deadlock? How do they get the brakes off?
Acclimatization again. It's all about adaptation over time. Over 2 to 3 days, the respiratory center in the brain loses its sensitivity to that low CO2, and it's the kidneys again.
The cleanup crew.
In this case, instead of adding bicarbonate to buffer acid, they do the opposite. They start to actively excrete bicarbonate into the urine. They are removing the base from the blood and, by extension, from the brain fluid.
So they're effectively resetting the thermostat. They're making the brain fluid slightly more acidic, or at least less basic, to compensate for the alkalosis from breathing hard.
Precisely. They cut the brakes. The brain stops inhibiting breathing due to the low CO2. Now, the low oxygen drive from the peripheral chemoreceptors is free to push ventilation as hard as it needs to. Instead of a measly 70% increase, the climber can now increase their alveolar ventilation to 400 or 500% of normal.
That is a huge difference. That's the difference between reaching the summit and passing out at base camp.
It is. It's why you have to climb high, sleep low, and take your time. You are literally waiting for your kidneys and brain to negotiate a new treaty on breathing control.
Moving on to section 6, the exercise mystery. I think this is my favorite part of the chapter because it completely, totally defies intuition.
It really does. It stumps medical students all the time.
The observation is simple. During strenuous exercise, your oxygen consumption and your CO2 formation can increase 20-fold. A massive metabolic demand.
Correct. The body is burning fuel like an absolute furnace.
So, logically, the expectation would be that your arterial oxygen should drop and your CO2 should rise, and that chemical change would trigger the heavy breathing. Cause and effect. Simple.
That is the logical assumption. That's what everyone thinks must happen. But if you put an athlete on a treadmill, insert an arterial line, and measure their blood during the exercise, the PaO2, the PO2, and the pH remain almost exactly normal.
They don't change.
They hardly budge. In a well-trained athlete, they are rock steady.
So if the chemicals aren't changing, what on earth is telling the lungs to pump 20 times harder? Where is the signal coming from?
This is where we have to look beyond chemistry and look at neurogenic control. The answer lies in the brain's command center. When the motor cortex in your brain transmits signals down to your muscles saying run, it simultaneously transmits collateral impulses to the brainstem respiratory center.
It's an anticipatory signal. It's like sending a CC on an email to the lungs.
Exactly. The brain doesn't wait for the blood to get dirty with CO2. It says, we are about to do heavy work. The muscles will need more oxygen and produce more CO2. Start the fans now. It's a feed-forward mechanism, not a feedback mechanism.
Figure 4210 illustrates this beautifully. It shows that when exercise begins, ventilation increases instantly.
Instantly, the very first second, before any chemical change could possibly travel from the muscles, through the veins, to the heart, to the lungs, and then to the brain. There's no time. In fact, sometimes the brain overshoots.
What do you mean?
The ventilation increases so fast at the very beginning of exercise that the arterial PCO2 actually drops slightly below normal for a moment because you're breathing too much for the current metabolic rate.
That proves it's not a reaction to high CO2. If it were a reaction, the CO2 would have to go up first, not down.
Precisely. And the text also mentions that this is partly a learned response. With repeated periods of exercise, the brain gets better and better at matching the breathing signal to the muscle signal. It essentially learns the proper game for the feed-forward loop to keep the blood gases perfectly stable.
So training isn't just about your muscles and your heart getting stronger. It's about your brainstem learning exactly how much air you need for a given sprint or a given climb.
Yes. It's optimizing the neural feed-forward loop. It's neural training just as much as it is muscular training.
That is incredible. The hidden autopilot is also a machine learning algorithm.
It really is. It refines its predictions with experience.
Okay, let's hit section 7. We've covered the main systems, but there are some other factors and some strange breathing patterns mentioned that we should touch on. First off, voluntary control. We can hold our breath. We can choose to breathe fast. We have some say in this.
We do. The conscious mind, the cortex, can override the autopilot in the brainstem. But only for a while. Eventually, the chemical drivers, specifically the buildup of PCO2 and the subsequent rise in hydrogen ions in the brain, will become so overwhelmingly strong that they will override your will and you will gasp for air.
So you can't hold your breath until you die?
You cannot. You will pass out first, and the moment you lose consciousness, the autopilot will kick back in and make you breathe.
Good safety feature. Then there are irritant receptors.
These are located in the lining of the trachea and bronchi. If you inhale dust, smoke, or even just cold, dry air, these receptors get irritated and trigger protective reflexes like coughing, sneezing, or even bronchial constriction. This is a key factor in things like asthma and emphysema. It's a defense mechanism.
What about these J receptors? That's an odd name.
J stands for juxtaposition. They are located right next to, in juxtaposition to, the pulmonary capillaries in the lungs. They get stimulated when those capillaries become engorged with blood or filled with fluid, like in congestive heart failure or pneumonia.
And what do they make you feel? What's the sensation?
Dyspnea, air hunger, that terrible subjective feeling of, I can't breathe, even if your blood gases are okay. It's not about chemistry. It's about a feeling of pressure or fullness in the lung tissue itself.
Now, a darker topic mentioned is brain edema and anesthesia.
This is critical for understanding respiratory failure in a clinical setting. If the brain swells, we call that edema, say from a head injury in a car accident or concussion, the skull is a fixed bony box. The pressure has nowhere to go but up.
Right.
This pressure can compress the blood vessels that supply the brainstem. This can block the blood supply to the respiratory center. The neurons shut down from lack of oxygen, and breathing stops.
The text mentions a temporary fix for this, intravenous hypertonic mannitol.
Yes. Mannitol is a sugar alcohol that can't cross the blood-brain barrier. When you inject it into the blood, it makes the blood very concentrated. By osmosis, it then pulls fluid out of the swollen brain tissue and into the bloodstream. It shrinks the swelling, relieving the pressure, and sometimes restarting breathing. It buys time for the neurosurgeon.
In anesthesia or narcotics, this is obviously very relevant to the opioid crisis.
This is the exact mechanism of death in an opioid overdose. Drugs like morphine, fentanyl, or even pentobarbital directly suppress the respiratory center. They make the neurons less sensitive to CO2. So the CO2 rises in the blood. but the brain doesn't respond. It doesn't send the ramp signal. Eventually, the drive becomes so depressed that the person just stops breathing.
It effectively turns off the autopilot.
It silences the entire system.
Section 8 discusses a very specific, strange breathing pattern called Cheyne-Stokes breathing. This is fascinating to watch clinically, but usually a very bad sign.
It is visually very distinct. Every medical student learns to spot this from the doorway of a patient's room. It's a periodic breathing pattern. The person takes a few deep breaths, then the breathing slowly wanes, gets shallower and shallower until it stops completely. That's the apnea phase.
So they just stop breathing for a bit.
For maybe 10 or 20 seconds. Then, after that pause, the breathing slowly waxes back up again, getting deeper and deeper until it peaks. The whole cycle repeats every 40 to 60 seconds.
It looks like a sine wave of breathing. What causes this oscillation? Why can't the system find a steady, stable state?
It's a classic instability in a feedback loop. Figure 4212 explains it well. Basically, the person overbreathes for a bit, which blows off too much CO2, lowering their blood CO2. The brain senses this low CO2 and says, whoa, stop breathing.
Okay, so they stop apnea.
But while they are stopped, CO2 produced by the body builds up again. Eventually, it gets high enough that the brain senses the high CO2 and restarts breathing. But it overshoots again. It breathes too hard, blows off too much CO2, and the cycle repeats.
Why doesn't this happen to everyone? My system seems to regulate smoothly.
Normally, the gas is dissolved in our body fluids, and tissues act as a massive buffer. They damp the signal so we don't oscillate wildly. Cheyne-Stokes happens when that damping fails, usually due to one of two main causes. Cause number one, a long delay in the transport of blood. This is very common in severe heart failure.
How does that delay cause the oscillation?
Imagine you are in a shower. You turn the knob to hot, but the pipes are really long, so the hot water takes 30 seconds to reach you.
So I stand there freezing, getting angry.
Right, so you turn it up more. Why isn't it hot? You crank it all the way to hot, suddenly, bam, scalding water hits you.
So I scream and turn it all the way to cold.
But the scalding water is still in the pipe. So you burn for 30 seconds before the cold finally hits you, then you're freezing again. That is Cheyne-Stokes in heart failure. The blood moves so slowly from the lungs to the brain that by the time the brain senses the gas levels, they have already changed drastically back in the lungs. It's regulating old news.
That is a perfect analogy. What is the second cause?
The second cause is increased negative feedback gain. This often happens with brain damage, like after a stroke. The brain essentially overreacts to small changes. Instead of a gentle proportional adjustment to the shower knob, the brain yanks it fully on or fully off in response to the tiniest temperature change. It's a hypersensitive switch.
So Cheyne-Stokes is effectively a control system oscillation caused by either a time lag or an oversensitive controller.
That's it, exactly.
Section 9 covers something much more common, something millions of people deal with: sleep apnea. The text says this can occur 300 to 500 times a night in some people.
Yes, apnea just means the absence of spontaneous breathing. And there are two main types. By far the most common is obstructive sleep apnea.
This is the plumbing issue, right? Not the wiring.
Correct. The muscles of the pharynx, the back of the throat, naturally relax during sleep. In some people, due to a narrow passage, extra fat deposits, or enlarged tonsils, that passage closes completely. It collapses.
So the signal to breathe is there. The dorsal group is firing its ramp signal, but the door is shut.
Exactly. The chest and diaphragm heave, but no air can get past the obstruction. The person snores loudly, then the airway collapses. Silence. During that silence, the oxygen in their blood drops and the CO2 spikes. This creates a sudden, violent chemical stimulation of the respiratory center.
And that wakes them up.
It causes a microarousal from sleep. The person makes a loud gasp or snort, which forces the airway open. They take a few deep breaths, their oxygen comes back up, and they fall back asleep. Then the cycle repeats. Hundreds of times a night.
And the consequences are severe. It's not just about being tired the next day.
No, it causes fragmented sleep and severe daytime drowsiness, absolutely. But physiologically, every time you have one of these gasping arousals, your sympathetic nervous system spikes. It's a fight or flight response. This leads to a high heart rate, high blood pressure, and over time, significant cardiovascular disease. It damages the heart and blood vessels.
The treatment mentioned is CPAP.
Continuous positive airway pressure. It's a machine that delivers mildly pressurized air through a mast to essentially splint the airway open with air pressure. It's like inflating a tire so it can't go flat.
Then there is the other type, central sleep apnea.
This is the wiring issue. Here, the neural drive from the brainstem to the respiratory muscles transiently ceases. This usually comes from damage to the respiratory centers themselves, like after a stroke or from an extreme sensitivity to sedatives or narcotics. The brain simply forgets to send the signal to breathe.
That is terrifying.
It is. Most cases of sleep apnea are obstructive or a mix of obstructive and central, but pure central apnea is much rarer, usually accounting for less than 1% of cases.
Finally, let's go to Section 10, the limits of the human machine. How long can a human hold their breath? The numbers here seem impossible.
Well, the text mentions world records of over 24 minutes.
24 minutes?
Yeah.
How is that physiologically possible? You just said the CO2 builds up and we are forced to breathe.
The setup is key. The elite competitors don't just jump in the pool and hold their breath. First, for several minutes, they hyperventilate with pure oxygen.
Okay, let's break the chemistry down. Why does that work?
Two things are happening. First, remember, the primary urge to breathe comes from high CO2, not low oxygen. Hyperventilation blows off massive amounts of CO2 from the body, so you start the breath hold with extremely low CO2 levels. This removes the urgent chemical signal to breathe for a very, very long time.
And the pure oxygen part.
That fills the lungs' storage capacity and, more importantly, saturates the blood and tissues with a huge reserve of oxygen. So you have plenty of fuel, the O2, and you've gotten rid of the waste product, the CO2, that normally triggers the alarm.
But there is a huge danger here for regular swimmers who might try this. Shallow water blackout.
Very real and very deadly danger, yes. If you do this, especially without the pure oxygen, just regular hyperventilation with air, you can lower your CO2 so much that you have no urge to breathe. But your oxygen is still being used up by your brain and muscles.
So you run out of oxygen before you ever feel the need to take a breath.
Precisely. You can black out from hypoxia, a lack of oxygen to the brain, without ever feeling that desperate air hunger. Unconsciousness usually hits when your oxygen saturation drops to around 50%. If you pass out underwater, well, the autopilot eventually tries to kick in, you inhale water, and you drown.
So the hidden autopilot can be tricked, but at great risk.
Yes. The system is designed to keep you safe under normal conditions, but if you deliberately manipulate the chemical variables, you can bypass the safety warnings with tragic consequences.
We have covered a massive amount of ground today. From the dorsal respiratory group setting that basic rhythm to the ramp signal smoothing it all out.
We looked at how CO2 disguises itself to cross the blood-brain barrier and become the primary driver of breathing, and how our kidneys help us adapt to altitude by manipulating bicarbonate.
We solved the mystery of exercise. It's not a reaction to bad blood gases, it's an anticipation, a neural feed-forward from the brain's motor cortex.
And we saw how delicate the balance is, looking at the wild oscillations of Cheyne-Stokes breathing and the nightly blockages of sleep apnea.
So what does this all mean for us? Why does a regular person need to know any of this?
I think it just highlights the sheer elegance of our own biology. We go through our days worrying about our emails and our to-do lists, while this incredibly complex silent control room in our brainstem is constantly sampling our blood chemistry, calculating ramp signals, and adjusting for exercise before we even take a single step. It's a reminder of the incredible machine we are all living in.
The final thought that really sticks with me is the central irony of it all. We are oxygen-breathing creatures. We will die without it in minutes. Yet our primary minute-to-minute drive, the thing that forces us to take that next breath, isn't the hunger for the oxygen we need. It's the need to get rid of the waste, the CO2.
It's a waste management system that, as a side effect, keeps us fueled.
I love that. Here's a provocation for you to take away from all this. Next time you exercise, maybe going for a run or just lifting something heavy, pay close attention to that very first breath you take as you start. Did you breathe because you were already out of air? Or did you breathe because your brain told you that you were about to be? Think about that signal. Thanks for listening to The Deep Dive.
See you next time.